Okay, so this little mini online lecture is to talk about translation. And we already looked at this slide in class today. So what I want to remind you is we're talking about the process of taking an mRNA sequence and translating it into a totally different language of proteins. And you will remember, because you watched the protein online lecture, that proteins are made of amino acids. Remember, all of this is happening on the ribosome. That is all taking place in the cytosol. Remember, all translation starts on a ribosome in the cytosol, and then it could move to the rough ER. So you remember this is from the endomembrane system. Okay. The other important thing to remember about translation is that it's 3 RNA nucleotides. We call that a codon, and they code for 1 amino acid. And the translator, the molecule that knows both languages, is tRNA. Okay. And that's shown here in purple. So tRNA we'll talk about again in a second, but tRNA has a anticodon that base pairs with the mRNA codon. And remember, whenever we're doing base pairing, things are always happening anti-parallel. Okay. So we always read our codons 5' to 3'. Oops. And because our anticodons are anti-parallel, they are read 3' to 5'. So you can see how they base pair. This tRNA structure here brings in an amino acid. That's what the little balls are representing. So what we didn't get a chance in class to talk about is how this actually happens on the ribosome. Keep with you your mRNA codon table. It's always written 5' to 3', left to right. Okay. It's the mRNA that we're reading. Yes, we talk about the anticodons base pairing to it, but we're always looking at the mRNA to determine the amino acids. So all these words represent the names of the amino acids, and we will be translating in 1-letter code because that's a little bit easier. We talked about the reading frame. So this is mRNA, and the reading frame is determined by the AUG start codon. Okay. And then every group of 3 is read until you come to a stop codon. Oh, I didn't know it did that. Okay, so back in this mRNA codon table, remember we have 3 stop codons, and AUG is always the start codon. And stop means stop. Even if there's RNA past that sequence, you stop translating at the stop codon. Okay, let's talk about the ribosome. The ribosome is made up of ribosomal RNA, rRNA, plus proteins. So the This is kind of a model, space-filling model of a ribosome. Here's a little bit easier schematic version, lets you see it a little bit better. One thing I want you to know is that the ribosomal RNA is the catalytic part of the ribosome. So what does that mean? That means the rRNA is the actual enzyme of the ribosome. And what does the enzyme do? It makes peptide bonds. So it connects an amino acid, I'm going to abbreviate that AA, to another amino acid, AA, and the peptide bond right there is a covalent bond. And I talk more about this on the video about protein structure, so make sure you watch it. Okay, so let's look at this a little bit closer. A ribosome has 3 sites called the EPA site. This in yellow here is the mRNA. Well, I guess it says that right there. So the ribosome, which is made up of a small and a large subunit, it's going to bind to the mRNA and it's going to read from 5' to 3'. And it's going to read in groups of 3, which are the codons. And remember, the molecule that reads the codons is the tRNA. So here you can see a polypeptide being made. Remember, a polypeptide is another term for a protein. So codons line up at the A site. So the A site is where the next codon is read. You can think of it as where the next amino acid comes in. The P site, as you can see, is where the peptide is being formed. And we're going to talk about, there's a transfer, you can't see that with my red. There's a transfer back and forth between the P and the A site for this peptide. Okay, and we'll talk about that in a minute. The E site stands for exit. So when a tRNA is brought or is moved into, when a tRNA is moved into the E site, the tRNA leaves, the tRNA exits. It leaves the ribosome. It exits. Okay, so let's look at the 3 stages of translation. So just like the 3 stages of transcription, you have initiation, you have start, you have elongation, which is grow, or you're forming the peptide, forming the polypeptide in this case. And you have termination, which is the stop or the end. So we're going to talk about each of these 3 stages. Eukaryotic translation initiation, so this is your first stage, is different than prokaryotes. Okay. So I want you to know how it works in your cells. So what happens is, remember we talked about a 5' cap and a 3' tail. And so for translation initiation, there are these what we call initiation factors that bind. Let me change my color here. So a bunch of proteins called initiation factors. Anytime you see the word factor, it means proteins. That binds to the 5' cap. And there are also proteins that bind to the 3' tail. And so initiation actually looks like this. You have your 5' cap, you have your poly A tail, and you have these cap binding proteins that bind here, and you have these other proteins in a that bind the poly A tail, and they have to interact together to bring on the ribosome. So I'm going to put the part of the ribosome right here. So we talked about the cap and the tail are important for translation, and they're important for translation initiation. So remember, they keep the mRNA stable, and they are important to get all the proteins there to start translation initiation. Okay, so we're not going to look at this part right now. We're going to focus up here, and we're going to leave the mRNA in a straight out model because it's a little bit easier to understand. So what happens, let me change, oops. It wouldn't let me, hold on. Go back to a better color. Okay. I don't want you. So what happens is this little purple part is the small ribosomal subunit. And this goofy looking guy is our initiator tRNA. And you know that all translation is going to happen at the AUG, so it's always going to start with methionine. So look at your codon table, and you'll see that methionine is AUG. That's the start codon. That's the first amino acid for all proteins. What happens, which is really interesting, is that this whole complex that's shown right here binds to the cap, like I was showing you, and then it scans the RNA molecule until it finds that AUG. So if you look back to our example here, see all of this? The ribosome, here's your cap, is going to bind here, and it's going to scan until it finds AUG. Once it finds AUG, it's ready to start translation. So at AUG, the large subunit comes on, large subunit comes, and the ribosome is fully formed. Okay, so we have our ribosome, we're going to talk about our EPA sites, and here's our AUG. So So this is initiation. The small subunit and some initiation factors, some proteins, and the initiator tRNA, the MET tRNA, form a complex, and they bind the cap, and they scan, so they're moving along like a train on a track until they find the right AUG. And what's happening at that AUG, here it is, our start codon, you have that initiator tRNA with its methionine amino acid hooked in. The large ribosomal subunit comes on, and now we are ready for translation elongation. One of the things you can see here is this is our tRNA, and you can see the anticodon. So now you can see how base pairing happens, A and U and A and U and C and G. This is 3' to 5'. The mRNA is 5' to 3'. So just like everything we've been talking about, whenever you're base pairing, it's 5' to 3'. That's how the nucleotides, the bases, can fit together. Okay, so I don't know if I can show you these videos. Let's see if I can show you and walk you through. Probably have to turn on my sound. So I write that this video is kind of ugly, but it's accurate. I can't remember if it has sound. But here's the tRNA. Maybe it doesn't have sound. With the methionine, and what it's trying to show you is it's scanning to that AUG. Okay. So here it goes. Hydrogen bonding, just like with DNA, occurs between the anticodon. Oh, this is bad. And then the big part of the ribosome, the large ribosomal subunit, comes on. So, not a beautiful image, but it's kind of trying to show you that scanning process. The reason I picked this animation is a lot of other animations are for bacterial translation initiation, and bacterial translation initiation does not scan. So that's why I had to kind of show you the ugly one. Okay, so we talked really briefly about tRNAs in class. tRNAs are what we call a non-coding RNA. So they don't encode a protein. They are functional as an RNA molecule. Molecule. Okay. They're encoded by genes. They're transcribed, but by a different RNA polymerase. They have what we call this clover leaf structure. Right up here at the 3' end is where the amino acid is bound, and each tRNA binds to a specific amino acid based on its anticodon. What does it need to bring in? I drew a blob over this, but if you can see, if you follow this around, this is the 3' end, this is the 5' end. Okay. So the anticodon is also 3 nucleotides of RNA, which is antiparallel, and base pairs to the mRNA codon. Okay. So we've talked about initiation, right? mRNA, first tRNA, ribosomal subunits. Elongation for translation is from start codon to stop codon. Remember that start codons and stop codons have no effect on transcription. They're only important for translation. And then what we'll talk about in a second is termination, where we reach the stop codon. So let's look at these stages. Translation, elongation. Would like to see if this works. If I can't, oh darn. Okay, so you have to have Adobe Flash Player. And apparently I don't have it on this computer. Let me just see something. Sometimes you have to go up to that little lock thing and hit allow. I don't know why it's such a pain in the tush. Okay, let's see if this will play and I can walk it through with you.
Chain elongation begins with the binding of a tRNA, which recognizes the next codon in the mRNA to the A site of the ribosome.
Okay, so what this is showing you is elongation. So we made a couple peptides. We've hooked a couple amino acids together. The codon that was just read is in the P site. The A site was empty, right? So I can back this up. The A site was empty, and now the tRNA is coming in to base pair with the new codon
Of the ribosome. This is catalyzed by the EFTU transcription factor and requires the hydrolysis of a GTP.
You don't need to know either.
Once the tRNA binds in the A site of the ribosome, the polypeptide chain is moved from the tRNA in the P site to the amino acid attached to the tRNA in the A site.
Okay, so remember I talked about how we have this transfer of the peptides back and forth between the P and the A site. Okay, so the P is where the peptide is housed, then it's transferred to the A site, and then you're going to see that this tRNA is moved to the P site.
Peptidyltransferase, a protein RNA complex present in the 50S ribosomal subunit, catalyzes the formation of this new peptide bond between the amino acids. The ribosome then translocates to the next codon.
Okay, so really important. The bond made between the amino acids is called a peptide bond. It's a nice, strong, covalent bond. And now the ribosome is going to move so it can read the next codon in the A site. And let's see what happens at the E site.
This process is promoted by elongation factor G and requires another GTP.
You don't have to know that.
This places the empty tRNA molecule in the E site of the ribosome and moves the tRNA containing the growing polypeptide chain in the P site. The next codon in the mRNA chain is positioned in the A site. The uncharged or empty tRNA in the E site then leaves the ribosome and a cycle of chain elongation is completed.
So
Through subsequent cycles of chain elongation, the polypeptide chain continues to elongate one amino acid at a time.
Okay, so we're going to go P site's where the peptide is made. A is where the new amino acid comes in. We connect them. The whole ribosome shifts down the RNA to read the next codon. The tRNA that's lost its amino acid is in the E site and exits. And we keep going and going. That's elongation. Okay, so this is just showing you the cycle. Peptide is made. New amino acid comes in. We have a transfer. Right, so we make a peptide bond. The ribosome shifts. This tRNA is now here, so I'm going to keep track of it like this with the X. Right. This tRNA with the green squiggle, that you can't see. This tRNA with a black X. Okay, we follow that. That gets ejected. That gets exited. Now we have an empty A site. A new tRNA is going to come in. You're going to just repeat and repeat and repeat until you reach the stop codon. Okay, well, so let's look at termination, and I'll walk you through this.
Termination begins when a stop codon appears in the A site. Since there is no tRNA corresponding to the stop codon, a release factor binds in the A site. The binding of the release factor...
Okay, so let me back up a little bit. That release factor is a protein. Okay, I'll write this down in a second. So there are no tRNAs that read stop codons. So if you look at your chart, you have 3 stop codons. There are no tRNAs that read that, because then it would have to have an amino acid. So the release factor comes in. It's a protein that kind of fits the shape of a tRNA, and you'll see what happens.
Site. The binding of the release factor causes the polypeptide chain to be cleaved from the tRNA.
That means cut.
The polypeptide is released, and then the tRNA is released. In the last step, the 2 ribosomal subunits and the mRNA dissociate from each other. This completes the termination process.
So what I want you to see is that the mRNA is intact. The mRNA can be translated over and over and over and over again. The ribosomal subunits are still intact too, so they can be reused over and over again. The polypeptide chain is now going to fold into its protein structure, and you've learned about primary, secondary, tertiary, and quaternary structure. Okay. The tRNAs will go and get a new amino acid hooked to them, and we'll do this whole process over and over again. So termination involves the stop codon, right? We have 1 of 3 in the A site. A protein comes in, and that's called, that protein is called a release factor. And the big important part here is it's not a tRNA. Protein comes in, the whole complex disassembles. So that's why I was telling you guys in class, stop means stop. You can't keep translating past that stop codon because the ribosome has fallen apart, the mRNA has been released, and you actually have to start over and go back to the cap. So, a couple clicker questions we didn't get a chance to go to. What I would suggest you do is you pause this slide, or this video. You have this in your handout from class, and you translate the following mRNA. Okay? You're going to have to find the start codon and the stop codon. So pause the video, translate, and see what you get. Okay, hopefully you've done this on your own, and as you're scanning, you find, AUG. Right? Now, you want to go break in your groups of 3, and I highly, highly, highly suggest you do this. Okay, and so you're going to start reading AUG, AGC, GAC, and I'm going to tell you the answer is C, because UGA, UGA, whoa, what happened? UGA is a stop codon. So even though there's one more codon here, you cannot translate it. The important thing too is you have to scan for the AUG. All right. This next question, it says which amino acid will be added next to the polypeptide chain? Okay. So what I want you to see is this is the mRNA, this is the tRNA, it's already brought in leucine. So what is going to be the next amino acid? So pause the video, figure it out, look at your codon chart. And hopefully you figured out the GCG codes for alanine. So the anticodon would be CGC 3' to 5', and that would be hooked to your tRNA with alanine amino acid. Okay, just a few more slides. Remember, you really want to be able to distinguish coding versus template strand. The easiest way to remember it is template is the one that RNA polymerase copies. And it has to copy by base pairing. Okay? So if you look, you've got a T here, you've got an A here, you've got a G here, you've got a C here. A, U, and it keeps going. And then if you were translating, you would come across that first AUG start codon. You would grow in your groups of 3 all the way to the stop codon. Here you see at the 5' and the 3' end you have some untranslated regions. That just means that that RNA is not copied into proteins, right? It's not translated, untranslated. So the untranslated region of the 5' is before the start codon. The untranslated region of the 3' is after the stop codon. And we don't get into it in this class, but this is all for control, right? So you need to have that 5' cap so the ribosome can bind and scan. The untranslated region is also important for part of the control of translation. You have this picture. Unfortunately for you, it's black and white because I can't copy in color. But again, it's trying to let you see the coding strand, the template strand, the mRNA, and then how translation happens. So, gene expression, or the central dogma, is DNA to RNA to protein. And these arrows stand for transcription and translation. So, heads up, the short answer question on your post-class quiz number 11 this weekend is to compare and contrast transcription and translation. So get that all figured out before you take the quiz. Hopefully you can see some differences, such as transcription is happening where? In the nucleus. Translation is happening in the cytoplasm or on the rough ER. And remember, we have a ribosome that's actually doing all of this. You've got tRNAs and mRNAs, and in the ribosome you have rRNAs. You're making proteins out here versus transcription, you're making RNA. You've got a lot of base pairing going on in both cases. So think about the similarities and differences between transcription and translation. Last topic I want to have you understand is what we call the redundancy of the genetic code. So if you've been looking at this mRNA codon table, you'll notice that many of the amino acids have multiple codons. In fact, serine has 6. So there are 6 different mRNA codon sequences that code for the one amino acid, serine. The start codon methionine, or the start codon AUG that encodes for methionine amino acid only has 1. And I think, oh yeah, tryptophan here that's represented as W, that's the only other codon, or I guess I should say amino acid with 1 codon. What this means is that you can have different DNA, which means different RNA sequences that encode for the same protein, right? So just looking at phenylalanine, your mRNA could be UUU or UUC, and you would get the same amino acid put in. So that's what we mean by redundancy of the genetic code. There are some kind of overlapping same information in the codons that code for the same amino acid. So, how I want you to try to see if you understand this is pause the video. And the question is, how many different mRNAs could code for the following peptide? So M is methionine, A is alanine. So you need to get out your genetic code and take a look. Okay, hopefully you came up with the answer 4. Because methionine only has one, AUG. But alanine, where's alanine? Right here, could be GCU, GCC, GCA, GCG. So AU, I already forgot it, GCU, or AUG, GCC, or AUG, well, I should write it right, GCG, or AUG, GCA. All of these encode for the same peptide, the same 2 amino acids, methionine and alanine. So that's an example of how the genetic code is redundant. All right, that concludes translation. Once you've studied this information, you should be ready for your quiz.